I'm excited to welcome you all today to our Clinical Core and Clinical Task Force session, which will be happening from 2 to 4 p.m. Eastern. So without further ado, I wanted to pass things off to Mary Sano from Mount Sinai ADRC. She is an ADRC Clinical Task Force member. So Mary, you can go ahead and take the floor.
Great, thank you. I'm gonna begin by sharing my screen. All right, well, welcome everybody. Thank you for being patient and welcome to the Clinical Core Steering Committee session. Second time we're doing this virtually. I just wanted to give you a brief rundown on what we're going to be covering today. I'll give you a little update on how the steering committee has adapted to this challenging time. And then we're going to have a great presentation from Howie Rosen describing what the job of clinical cores might be in helping communities address how to assess dementia. And then finally, we'll just give you a minute to explain the relationship between the clinical core and the clinical task force. So it's been a long and winding road. And I have to say, as I began my 1 year in this position, I was thinking about how COVID really changed our lives. We've spent so much time thinking about this disease, about testing, about vaccination. And as today comes around, we hear that we may be moving to a truly improved state. I wanted to take a minute to remind you what Clinical Core Steering Committee is supposed to do. We basically set the agenda for the Clinical Core meeting. We have monthly meetings to do this. The NIA attends. We provide regular feedback to NACC and NIA and to core programs to really improve the in-person meetings and the interactions among the centers. I think we've been really successful, and I'll describe some of the things that we've done in the past year. Members of the Clinical Core Steering Committee also serve on the task force, and you'll hear about that. And we also have a responsibility to show a commitment and follow the important interest groups to be sure that we engage wide communities, including those who are typically underserved. In addition, the committee is asked to set or achieve a few major goals during their term. And these can be chosen from the recommendations to the ADC from its advisory group. And this is just a list of 3 of the recommendations that came to the clinical core. One was to address gaps in disease mechanisms and risks. Another, clinical research capacities, being sure that we build them both for ourselves in research as well as in the community. And a discussion of translational research. How can we be sure that we maximize the translation of what we collect by working with our other cores? The Clinical Core Steering Committee has in the past put together a really great program for this session. But as you know, we have really important work of the task force telling us what new approaches we may be considering for our evaluation in the ADCs. So what our committee did was they took the important topics that had been presented to us from center directors across the whole network of NACC and created a series of webinars. And here's a listing of the webinars that have happened in the past year. We alternated our monthly meetings and then presented these webinars, which were well received and now are posted so that the information is available. And as you can see, it continues to be viewed by many people. So we're really quite proud of the fact that we were able to continue the work despite the real challenges of this COVID period. Now, in choosing what we would like the clinical cores to think about, we came to realize that the clinical core gathers incredible knowledge from the community through the UDS. We have standardized assessments of demographics, some medical information, a targeted medical history. We have standardized cognition and neurological assessments. And there's a huge number of publications that have come out of this work. And the question we ask is, how can we translate this for use by the community practitioner? So as the potential for approval of new treatments approach, a lot of attention will be paid to how dementia evaluations can be conducted in the community. And the question we wanted you to think about is, can and should we take a role in supporting the PCP to make a diagnosis of dementia with confidence? And so with that, I want to introduce Howie Rosen, who will present some information on this topic. And we realize that we don't have the perfect venue for a dialogue, but we will try to share information that comes to the Q&A and the chat and see if we can get some feedback about how you feel about this important topic. So Howie, please take it away.
Thank you, Mary. Can you hear me okay? Is that it? Am I being heard?
We can hear you, Howie.
Excellent, thanks. Just wanted to make sure. Okay, well, thanks again, Mary, for asking me to talk about this. I share your enthusiasm for us talking about this. I think I think Mary's intent was really, as she said, to open up a dialogue and not to make assumptions about the role that we should take in this activity, but rather to solicit the opinions of the group as to whether we should take a role that's different than we have now, and how much and what that should look like. So that's the intent of this talk, is to promote a dialogue using some examples to just illustrate where I think things have been, and to spark some discussion. So I'm going to be talking a little bit about the potential role of the clinical cores in community assessment of dementia. And I think I'm going to be highlighting work that has been going on for a long time, and I think is expanding, but hasn't really been a major focus of discussion traditionally in the clinical core environment. So this is, certainly preaching to the choir, and this is work, data from the Alzheimer Association showing how, assessment of dementia in community settings and cognition is relatively not attended to relative to assessment for other kinds of common medical risks. I think we're all aware of this. And I think, just as an example, when I speak to my colleagues, here's a quote from a very thoughtful and close colleague of mine who, said, well, this is how I approach it. If a patient has a complaint, I offer them a referral to the MACC, the Memory and Aging Center at UCSF. If they don't want to do that, I do a MOCA. And if that's abnormal, I start ARICEP. And I'm sure most of us have heard some version of this kind of approach in clinical practice. And of course, we all agree that that has limitations. And for instance, the MOCA is not very sensitive for MCI. It's not equally sensitive to all types of dementia. It may not work well in diverse populations in various languages and cultures. And so even for a primary care practitioner in the community to apply that logic that my colleague said, they would actually have to be more expert than they likely are in terms of even how to pick an alternative to the MOCA in the right setting. And that's just one example. And so to kind of further the discussion in that area, I'll point out that there is work being done by people who are part of and related to this community. And one example is the Detect CID study, which is a NINDS funded study that is primarily being led by by these three centers, Northwestern, Albert Einstein, UCSF, each of whom has their own cognitive assessment tool that they've developed, which is meant to be fast and sensitive and versatile, i.e. useful in diverse settings and populations. And so this is an example of a study that's tried to address this detection issue that is mostly happening outside of the specific context of the clinical core, and that may be fine. But of course, then we have the issue of, what if somebody detects a cognitive impairment that now needs a more detailed workup to us. So what happens then? So, will they make referral to specialists like our groups are with our clinical hats on, as opposed to research hats? Of course they might, and they often do. But I'll point out that, if detection went up, as I'm illustrating here, there's a lot of data that shows that might quickly overwhelm our workforce. I'll point out that for many of our centers, waiting lists for evaluations are already months long. We're doing our best to reduce that, but that's already in the context of a relatively low rate of detection. So if it goes up, we're going to have to deal with that again. And if there's a treatment available, then that has impact for how early and quickly those patients get access to treatment if we become the gatekeepers. So, so if the idea is that, well, primary care practitioners can make these assessments and do a full clinical assessment. Well, here's more data on the left here also from the Alzheimer Association surveys that show that, they would agree that this is important to make these assessments. So what prevents them from doing it? Well, there are lots of barriers. I only list a few here, but they include, they're not comfortable. It's complicated. They're not sure they have the expertise to deal with all the nuances. They perceive many patients and families may be resistant to an initial assessment. They don't have time, and they also feel like, well, once I make the assessment, I don't know how to provide them with resources. These are just examples, many of which we've heard. I'm highlighting these two in yellow because we're going to talk a little bit more about them. So thinking about this, one of the things that we did in California over the last few years is create a toolkit, which is basically a diagnostic toolkit that's meant to help the primary care practitioner to make a full assessment of dementia, accounting for the fact that they have less comfort and less expertise. So we basically laid out, these are the questions we ask as experts. These are how we tend to interpret answers that we hear often. It tries to, it stresses differential diagnosis and basically takes the approach that if it seems like Alzheimer's disease and all the questions and assessments point in that direction, that might not need a referral. But if it's something atypical, then it should be. And we try to provide a lot of guidance as to which cases should be referred. We give scripts to help with disclosure, et cetera. So the point is we developed this toolkit. This was developed really as a shared effort of the California centers in their role as state centers. But of course, you'll notice that many of these are also federally funded Alzheimer's centers, the majority of them. So, so when we did this, we thought, well, here it is. Now we created this toolkit. Here you go, primary care practitioners. Now you could do things. But of course, we learned that they still perceive it as very, very long, and they don't really know how to work it into their practice. And, we'll get this common response. I have 20 minutes to do an assessment. There's a lot of other things on my plate. I need something that'll take 5 minutes. And we know that a full assessment can't be done in 5 minutes. So we were fortunate enough to have the opportunity to apply for some more funding through the state, where we now are doing a project with 3 primary care practices in California, in Chinatown, in SF, and Fresno, and a large center in Los Angeles. And this is in collaboration with some colleagues from the state network, where we're actually helping the primary care practices to figure out how to do this. How do they do the screening assessment? What do they do after? Which members of their practice would administer which parts? And eventually, we also are interested in some ability to get at accuracy, which, as you can imagine, is complicated, and we really can't look at all the research questions that relate to accuracy in the first pass. But we're doing what I think we can with this project. So that's happening. And in parallel, another example of something that pertains to this is Project Echo for Dementia. We're collaborating with the Alzheimer Association to create this virtual recurring conference, which is short didactics combined with cases that primary care practitioners bring. It's a very common model not in dementia, in other medical fields, particularly in GI, but the Alzheimer Association has really been spearheading, trying to expand it into dementia. And I'll note a couple of other ADCs are involved in this too. why I bring it up is it's not just an educational venue. It's a venue for directly supporting primary care practitioners in implementing the principles that we talk about. So it's another opportunity to work with them and potentially change their practice rather than just doing lectures and then hoping that that ultimately results in change in practice. So that kind of thing is happening too. And, my hope is that work like that becomes kind of the basis for a stronger relationship between our 10 state centers in California and the primary care practices that are in their catchment areas and environment. And we're trying to seek some funding to start that process using ECHO as a kind of a core activity that organizes those relationships and expands them. But if that relationship does expand, it's a new network that can potentially be utilized to do more research and implementation of diagnosis and assessment in community settings. And I'll point out that we're not the only state with centers like that, with networks like this. The Alzheimer's, the Wisconsin Alzheimer's Institute, and has been around for a long time. And I think it's more recently that Georgia set up a memory assessment network that is similar in concept. And colleagues from ours from the CADCs or from the federal ADCs are involved in these. And we have other partners. So there's a newly established BOLD CDC centers that also have the goal of increasing assessment, and we have to think beyond just recognition of dementia in the community. And that's also led by colleagues who are part of the ADC community, in particular Josh from NYU and his colleague is Sue Borson. So I think these were just examples to sort of say there are things going on that are very, very important. They speak to our issues of diversity and the fact that really the maximum diversity is in the community, and then many of our issues with diversity are having not as strong links all the time as we want to community settings. So are we missing opportunities for collaboration with each other by not thinking about this, for utilization of NIA resources or other federal resources that the NIA can mediate discussions about? And this is really, should we think of this as a topic for extended discussion at a future meeting? And then, when we think about that, I think we have to think about, look, what role should we play? Should we develop tools? Is that our role? We have to remember that those tools will have to evolve as biomarkers develop, and so who better than us to help decide what the community tools should look like when there's a blood biomarker that's widely available? And then do we have to test the tools? If we don't test them in the community, then, there's threats to their validity that aren't really assessed. And I think as Mary has noted when we talked about there's really a combination of questions that are, kind of traditional scientific things about sensitivity, specificity, validity, but also questions about logistics, just how does it done, which traditionally we think of as health services research, but they can't be separated so well because they both have to happen in the same environment. And without one, you can't really ask the other. So where does our role stop? And what if we think our role has to stop at a certain point, what partnerships do we have to identify to make sure that the rest of the job gets done? So that's really the whole talk. And it's really meant to kind of facilitate a discussion about whether this is something we should continue to focus on, how we should do it. And, hopefully I left a little time for us to talk about that. And I guess we should identify ways to get additional feedback to keep the dialogue going. So it looks like I might have left a few minutes.
That's great. Actually, we have 1 or 2 minutes. And one of the things I did want to say is that we have a few questions I want to read out, comments, because I think I'd like to have as many of the directors think about this and give feedback. We can give it through the to NIA through our listserv, and as well as other ways. But one of the comments made by Suzanne Kraft is that this is really important. And the question is, does our, do these methodologies also target MCI as well as dementia? So important because new treatments are looking in that direction to the very earliest stages of disease. So.
Well, it's almost a rhetorical question in the sense of the answer has to be yes. I guess I'll point out in our experience in the state, and there may be some of the people who were in that discussion, it was controversial in the sense that, you know, it's a little harder to come across a definition, and how important is it that cognitive testing be part of that? And there was some hesitancy. But then when we rolled out the toolkit in our project, so we're now using a different version that we thought had to incorporate something akin to MCI, because actually otherwise practitioners might not realize there's an in-between choice. And we have people basically diagnosing people with dementia that really were MCI, and we didn't give them that choice of saying, you know, there is a way to temporize and to put this complaint in a context that our field understands. So absolutely, I think it's mandatory, but how to do it in a way that's implementable in the community is its own question.
Great. All right. Well, I don't see more comments here, but I'm really hoping that we'll hear from members of ADCs all over to tell us exactly how they would like to participate in sharing what we know with the community. So, and now I just want to return to one last topic before we hand it over to the task force. Let me see if I can share my screen. And all right. So just a final reminder for those of us who are on these task force, we know exactly how we're related because we meet almost 24/7 or so it seems. But in fact, there is a method to that madness. The overlap of membership really ensures that we can integrate the experience in the clinical core with the task force's desire to find the best outcome measures. And I just wanted to talk about the success of that overlap before I turn it over to Allan Levey. Because of the close relationship, the clinical core was really able to support the rapid initiation of new protocols in the COVID period. For example, the COVID survey, the telephone assessment of cognition, and we've even developed a remote assessment of the neurological exam. We've begun the process of assessing by video, and we've introduced the concept of assessing people at alternate times, not just fitting everything into an annual assessment, but asking as needed on a once in a while basis. And perhaps the most important thing we've done is today we really abbreviated the session so that the real important work of the clinical task force can be presented to you. And so with that, I'm actually going to turn it over to Allan and let him describe the important work that the task force has to share with us.
Thanks, so Mary. That's great. Thank you. If we could unshare, we'll pull up some slides, but that was a perfect segue. Great introduction by you and Howie both. So the clinical task force has been meeting nearly 24/7, as Mary said, and we are actually pleased to do it. I've never had the privilege of meeting with such an invested, energetic, powerful group of people as this group. It's really been impressive. Just give me a thumbs up if you can see my slides okay.
You need to switch it.
Okay. Thank you. How's that?
Perfect.
I'm just going to give a brief background, and then you're going to hear from our three work groups, predominantly today from Cindy and Greg, who have done the heavy lifting over the last several months, and we're going to spend most of the time on that. But you're also going to hear from Rhoda and Jeff from the technology work group, and Andy and Lisa from the neuropsychology battery work group. We really have one overriding goal today, and that's to get your feedback. And so I can't impress upon all of you enough that we view ourselves as representatives of the community. We're not at all trying to come up with a new UDS4 that is ours, this task force is making. It's really one that's meant to work for the entire community as best possible going forward for the years ahead. Just to remind you who are the members of this task force that have done the great work. I want to thank Dr. Maslia for the certificate of appreciation. I think that's great. I think the rest of these folks are waiting for their big paychecks still in the mail. But this group really has been dedicated. I can't tell you how often we do meet. I was afraid to count the meetings we've been having. There's the clinical measures and diagnosis working group, the cognitive working group, and then the technology work group. And the committee membership, some of these are direct members from the clinical core steering committee, as you just heard from Mary. Others are appointed by the NIA. But everybody has been involved, and I think what we've all seen is a phenomenal amount of work done by the cognitive working group as COVID hit last year. They really did the heavy lifting to bring us the remote assessment tools. We're now in a phase where the clinical measures and diagnosis working group is doing a lot of heavy lifting, which we're going to talk about today. And I view the technology work group as much of their work is coming in the future. And just to put in perspective, what we've been hearing about, as we all know, the huge infusion of resources to the Alzheimer community that Dr. Hodes was talking about this morning, we've all benefited from. And it's all targeted around the 5 goals of the US National Plan. And when you think about our responsibility and our opportunity as the ADCs and the clinical cores, we really play a key role in so many aspects of the national plan. Of course, we all are aimed at preventing and effectively treating Alzheimer's disease by 2025. But as we just heard, we also are playing important roles in developing new care models that are going to improve clinical care and be necessary for the translation, including caregiving research. And we are, in many ways, one of the most essential elements for public awareness and engagement in our own communities. Our job is less to track progress, but to make the progress. And the national plan is informed, as we all know, by the summits that occur. We just went through the Alzheimer's Disease Research Summit last week. I hope all of you heard Lisa Barnes' really phenomenal keynote presentation. The plenary presentation on diversity was spectacular, I thought, and so nicely sets the stage for something that's a priority for all of us. And it really, the whole process of the summit is to highlight some of the progress, which is staggering, as we've seen, but more importantly, identify the gaps and opportunities that can inform the national plan, where the priorities have to be reset. And we heard resoundingly from the summit, as we've heard today in other sessions, that more and more there's a need for inclusion of diverse populations in the research portfolios. We need larger sample sizes, as we've just heard beautifully from the artificial intelligence sessions in the scientific session. It's the size of the data, the quality of the data are both absolutely essential to inform the future. And the more and more we can provide comprehensive phenotyping, all of that is really necessary to deconvolute the complex heterogeneous disease of Alzheimer's disease and related dementias. And then, of course, our goal is to get this out into the clinics through the translation. Again, something that we all know, but just to remind us, there's a lot of work for us to do, and the Alzheimer's centers play a key role. We've been focusing on, as a field, the biomarkers and amyloid and tau for the last decade or more. We've heard from the genetics community that we're underpowered still. I think Dr. Maslia has said the ADSP is aiming for 100,000 or so subjects by 2025. We know the whole genome studies have been underpowered to date. And we already have 40 or so genes, but still the majority of the variants of the heritability is still not understood. So we have a lot of genetics work, which is dependent upon larger sample sizes as a foundational work. We've heard, from many of the groups, including a couple of studies I've highlighted here from the Rush ADC, that there's a lot more going on that contributes to the cognitive decline that occurs in our patients other than Alzheimer's disease pathology. And so if you take in their studies from the Ross map cohort, if you look at Alzheimer's pathology, cerebrovascular pathology, and Lewy body pathology, collectively, these also account for less than the majority of the variance of what contributes to cognitive decline. And also from the Rush group, I think they've beautifully illustrated in this publication of theirs, the individual trajectories that people go through during the course of life. And so each one of these spaghetti plots is a different individual from the cohorts. And you can just see the rainbow of colors, which reflects the diversity of humans and what personalized medicine and precision medicine is all about. How can we identify people early on and better understand and predict their trajectories and of course intervene? A lot of the resources have not only gone to individual investigators who have been doing really creative work, but we've also seen the NIA really invest in the infrastructure to help the research community globally. This is a slide courtesy of Susanna Petachenka that she provided that I modified a little bit. But we know that there's a very robust translational pipeline that's growing. We heard about the growing number of clinical trials today. We know there are early programs like AMP-AD and the Treat AD centers, as well as programs that are very well known to the clinical cores like ADNI, ACTC, and the like. But what I modified here, when I look at this pipeline, my perspective is that the Alzheimer's disease research centers are in many ways the force that really moves things forward across so much of this pipeline. And I think our role as Alzheimer's disease research centers, and particularly the clinical cores and the task force, is growing given the calls for more diversity, larger sample sizes, and more complex phenotyping. So I think the centers are only going to grow in importance. So I think just for those who are new to the centers, as the center program grows or new to the field or just entering, thought a little bit of historical perspective about what the clinical task force is and why it emerged may be helpful. Certainly was for me thinking about it. The Alzheimer's Centers program was started in the NIA in 1984. That was the same year, for those who don't know, that Glenner and Wong purified A beta and identified its sequence. The NACC, the National Coordinating Center, was formed in 1999 with what was initially called the minimal data set. And this was largely to put together retrospective data that had been collected at the initial centers during that period. And then the clinical task force was really formed here in 2002. And it was, the vision for this was to help facilitate multi-center collaborations, begin this journey towards assembling larger sample sizes. And at this point in the field, it was absolutely critical to begin to understand the natural history of aging, with largely a focus on MCI. Defining what it means was a major accomplishment, which wasn't trivial. Redefining the sort of the older McCann criteria from 1984 of even defining dementia. And so much of this decade, I think the center has contributed to. The first uniform data set then was established in 2005 to help plan for the longitudinal data that would be collected in a more uniform way. And then there was a very soon after that, a revision, which was fairly minor. And then it was only in 2015 when this UDS3 that we're all using now was established. And for those who don't remember, the major revisions to the UDS3 were updating the neuropsychology. There's concerns that many of the instruments were proprietary, and we needed to get them out in the open for open science and transparency. There were some new instruments added around visuospatial assessments. The neuroexam was added. And interestingly, we took out the Hachinsky and UPDRS at that time to try to streamline this. So that was in 2015. If you think about what was going on in the field during that journey, it was in the early 2000s when the first PIB imaging agent was developed at Pittsburgh. We saw the formation of DIAN, the autosomal dominant network, the great work that led to our insights that there is disease pathology beginning decades prior to symptoms. Similarly, ADNI began to have a major impact, showing the same is true in sporadic forms of Alzheimer's disease. And if you look at the treatment trajectory down here on the bottom row that I put, of course, we have donepezil, and then memantine was the last FDA-approved drug in 2003. And we've seen, these wonderful reviews from Jeff Cummings every year and his group, giving us a comprehensive review of what's out in the field. And if you look at this whole journey over the last, 15 years or 20 years has really been sort of the amyloid era because of this natural history of understanding that. And now we know that there's much more. And that led to the formation of the AMP-AD as one of the outcomes of one of the research summits back in 2014 to use systems biology to begin to understand more of the complex biology of the disease and to use data-driven approaches to deconvolute it and identify new targets. And so it was in 2017 when there was a review of the external committee to give perspective to the NIA about what the centers should be doing. Mary alluded to some of these because they were relevant to the clinical core, so I won't go through them, but there was a very clear charge from the external review to streamline as much as possible the UDS that is taking a lot of time, and that the center should enable more individualized research that could be creative to help move the field forward by streamlining and taking away some of the burden. Secondly, to enhance the deep phenotyping that we've talked about, including not just the clinical and cognitive things, but to expand behavioral imaging, functional scales, and also biomarkers, all of which we well know are on the path at great rate. There's also a charge to consider neuropsychological tools that will better address mixed dementias, the endophenotypes that we talked about, and the heterogeneity that we now know underlies dementia. And then also, there's going to be increasingly a need to integrate electronic tools and web-enabled approaches to also begin to integrate with medical records to go towards digital biomarkers that we're going to be talking more about as well, and then periodically reevaluate. And so here we were after the external review, the streamlining committee was formed after that. That really gave us and the NIA a charge then. We launched the UDS4 Clinical Task Force, a completely new task force, in October of 2019. And shortly after starting that, we realized that the task force was charged for the reasons we just talked about to start addressing some of these gaps and opportunities that were also highlighted in the summit. One great example that was recently published from the Wisconsin group and the NACC team shows you the power of the large data collection. So what might seem as sort of simple questions like, are the trajectories of dementia different in Blacks and whites, can only really be answered well when you have large numbers. And this was a good example, I thought, of how the NACC data could be used effectively to show what was somewhat surprising, I think, to many, that the trajectory for Blacks on average is actually slower once dementia begins, despite what we know is a much higher incidence and prevalence. Then our favorite occurrence, the roadblock, occurred with COVID. And so this committee was really just getting going when COVID hit. And so much of the last year has been the pivot. As I mentioned, our cognitive working group just deserves enormous credit for the work they did getting us all up and going as soon as possible with remote assessments. And now it's time for us to go look down the road past that roadblock. We've established guidelines and principles that we've presented to you before. For the sake of time, I'm not going to go through them all. We do want to maintain the value of the legacy data. It's a given. There's a ton of data there that is valuable. We clearly heard the charge to minimize the participant and center burden. And so that is one of the things we need your help with, probably one of our greatest challenges as we go forward. We certainly all recognize the need to do a much better job at being inclusive in our study populations and making sure the instruments are valuable and applicable to all. And we also thought it would be very important that, you know, the centers should continue to be nimble, but before adopting things that are recommended for center-wide use for really uniform assessment, we should be thoughtful and use well-validated approaches as instruments that we can validate in crosswalk studies and multi-center studies with smaller groups of centers to make sure the new tasks are really robust as we had hoped. So there's a wonderful model. We call this the Jicha model, the atomic model of the UDS, which is shown here. It's been a very helpful icon for us to think about the UDS. We think about the UDS as the nucleus of the Alzheimer's centers, or at least the clinical evaluations. You know, it's broken up into helping us uniformly assess across the centers who the participants are, what their risks and comorbidities are, their cognitive profile, and of course, how we use these data to come to our best research diagnosis. And the UDS, of course, is the common measurements, but every center is obviously doing their own sets of assessments in addition. And so this is the balance, how to keep the core as small as possible to really allow the centers to be as creative and move the field forward, you know, as fast as possible. So that's obviously the biggest challenge. But as we recognize now, the uniform data set was really developed initially with a perspective that was fairly limited to the amyloid perspective of amyloid being a disease of plaques and tangles, where we now recognize dementia is clearly a heterogeneous disease with mixed etiologies that we need to be cognizant of. And we also, I think, sometimes don't appreciate how important the UDS has become to supporting so many of the large consortia that are really doing a phenomenal job advancing the field. From ADNI to Diane, Mark BCID, we've heard about yesterday, is doing great work, leads us the Young Onset Dementia Consortium, the DLB Consortium, and then all FTD. And of course, there are others that I don't need to name. But what people may not recognize is the UDS becomes the data set that allows studies to begin to be harmonized across, having larger data sets pulled out of different studies, and to make recruitment into these studies easier when they come from our centers. You can identify subjects through the UDS and funnel them into leads or Diane, et cetera. And as we grow our centers, these become the essential participants that are going to inform the genetics and biomarker studies of the future. And so trying to balance all that, the task force this afternoon would like to give you an update, and that's what we'll be doing. I'm going to turn it over to Greg and Cindy in a minute. But again, my main goal is to make sure you recognize that we're not delivering you and trying to pretend to deliver to you a work that's finished and a work that's far from perfect. And we want your input to help make it better. And these are all still in draft. So we've laid out a process with NACC to develop feedback, and we're here today at step one, where we want to show you updates of what is being considered, get your feedback, and then at the end of the breakouts today, I will just give a very brief description of where we're going and the journey and how to get more feedback down the road from you. So with that, let me turn this over to Greg and Cindy, who are going to tell you about the hard work the clinical task force has been doing, the clinical measures group.
Fantastic, Allan. That was just a great overview, and you have been a stellar lead for this group. I did want to start on behalf of Cindy and myself, who are leading the CTF Clinical Measures and Diagnosis Working Group, by thanking Mary Sano first and foremost for her decades of leadership, really across the UDS trip that you've taken us through, Allan, but also her service on the Clinical Core Steering Committee, and recognizing that discussions of the work that the Clinical Core Steering Committee has been doing in regards to the CTF task force really needed this platform to help engage input, because as Allan said, we are not dictating a UDS4. We really want everybody's input. We consider ourselves stewards of the broader ADRC network. Allan, of course, Cindy and I want to thank you for your leadership and clarity of thought in moving this forward, as well as all of the CTF members, not just within clinical diagnosis working measures and diagnosis working group, but within the cognitive and the technology work groups. It's been a tremendous effort. Of course, Nina, Cerise, and Grayson, especially for her administrative support and the support of the NIA in pushing for this change for us, which we need to not only meet current needs, but also with a vision to the future that is critical for us. And NACC has been incredibly accommodating, supportive, and have provided really the legs that we stand on. I think that's important, but most importantly are all of you, the ADRC network. I want to thank you all on behalf of myself and Cindy for entrusting us with the opportunity to represent you in trying to move some of these ideas forward. You'll have an opportunity later to meet the clinical measures and diagnostic working group members, as well as members of the cognitive and technology work groups in the breakout session. So again, our charge is very simple: to take a look at where we are and really to see how are we up to date and where are we going? What's happening in the next few years with those goals of minimizing site and participant burden to the degree that we are able, but really science is driving everything that we do. So our guiding principles here are not to, and we're not here today to present a final document by any means. We're here to present preliminary recommendations only, so that need to be fully vetted. And so I hope you all stay on, engage in the breakout rooms, the other activities that we have planned, because the future, the UDS 4.0, is in your hands. And this is a collaborative effort. Nobody summits Mount Everest alone. We always work as a team, and so that's critically important. Is this an impossible task? Yeah, you know, some might say, an impossible task, and it's all fun and games until somebody brings COVID to the base camp, and then we're left with a boatload of trouble. But I think we can make it there. And we're working through COVID slowly as a nation. We've had good news, promising news. We've been able to deal with it even while it's been in our presence, and we can move on. So the clinical measures and diagnosis group was really tasked with looking at these forms that are shown on your screen. And there are some of them that remain works in progress, and all of them are works in progress, but some more so than others. The CDR, the MPIQ, the GDS, and the FAQ have really been kind of sidelined, and we've had discussions on these, but we're really going to focus on many of the other forms because these forms remain to be fully vetted. And we're looking at data actually from NACC. What have we acquired from these forms? How has that data been used? What is useful? Are there other measures that could potentially reduce participant and site burden that could be and not sacrifice and or advance science in the future? We've had significant discussions on CDR versus the QDRS or other measures. As of yet, no decisions have been made. Certainly when it comes to looking at the NPI and the GDS, we do find that our own committees are lacking the breadth of experience that we need. And so this is really a call out to those geriatric psychiatrists or those with specialty expertise from other areas in the area of behavioral and psychiatric symptoms to please reach out to us. We need your input. Make sure you have the time to devote, though, as Allan said. You know, this has been a time intensive process for us to work through things. So how we approach this is of particular importance, I think. We actually set about by modifying the NIH review style template and actually giving that out to all the clinical measures and diagnosis work group and assigning reviewers the same way that we would do to let's take a look. Let's actually review the UDS forms and see where things are. Now, a little bit different from an NIH style review was not only a critique, but what we're not allowed to do at NIH style reviews is actually propose some recommendations here. Here's what might make this better. And they became the items for discussion in our bi-weekly meetings that were occurring. So it's been a long time. I didn't realize it's been a year and a half, almost 2 years now that we've been working on this, and we're getting close. And so today we're going to talk about some of the forms that we would like to review with you all. You should have all received these from NACC with highlighted kind of proposed changes. Everyone remember, this is a work in progress. That's why we didn't present you final forms and just say, this is the judgment of the committee. We're stewards for you and want your input. So we'll kind of walk through these anyway, and NACC really, again, has prepared these forms for us. If you've not received those, barrage NACC right now with a slew of emails. You should have all received them. Your leads in the breakout sections should have these on hand. So I'm going to walk through the first few forms and then hand this over to Cindy, my co-chair, who's been a delight to work with anyway, to walk through a few more, and then we'll close out and move on to the cognitive working group and the technology working group. Of course, beginning with Form A1 doesn't give it an importance over any of the other forms. I think, you know, there have been some considerations, not just about any item changes within the forms, but the instructions itself. And we are thinking, you know, COVID and certainly the telephone battery has enabled us to function in a new way that we haven't functioned in before. And when we think about some of these forms, the mode of administration, reducing on-site burden, while collecting relevant information is important. And so the instructions in italics, gather this information, whatever is best for your site. We don't think these are simple questions, whether they're answered over the telephone or actually in clinic, should be irrelevant to their meaning. And hopefully this will be helpful for folks. Of course, there are some proposed changes to the questions. And one of them was, we have multiple, what's your primary race, your secondary race, your tertiary race, other races. And the US Census doesn't operate this way. We thought that we might be able to combine these all into a central question, which is a list of race, ethnicity questions, and check all that apply. This also came up because there were some considerations and comments from folks who might have equally mixed racial or ethnic backgrounds. Asking them to select a primary is like picking which one you like best, mom or dad, and that may be less than inappropriate. And we will still capture the information that we need here to complete, obviously, our demographic forms for grant applications and or progress reports and updates on cohort status. Thinking about LGBTQ as well, what we're really interested in is sex at birth rather than gender identity per se at the given time, believing, or at least, where the science is telling us that there is a component of perhaps hormonal status, the X versus Y chromosome difference that may play a role here. And this may help clarify and hopefully not put our patients or our participants in an awkward position of understanding, I might have been born with this sex, but I'm not that now, or have changed otherwise. We probably could do more in the future in terms of actually capturing current sex and or gender identity. That's open for discussion. Marital status and residence were distributed across multiple questions that were somewhat redundant. We've condensed these into a single question. Functional dependence was assessed, but we have the FAQ for that, or whatever might take the place of the FAQ. This is redundant. Let's try to streamline this form. And there was a push for adding occupation. This has come across in multiple of the affiliated U19s and was highly pushed by our colleagues from the biostatistics data management groups input as well. We're looking at the health and retirement study, occupational coding. This is potentially a significant social determinant of health. And we did think that in the core UDS, there are many centers that are collecting social determinants of health, but that there should be a small core set of critical questions. And I worked quite closely with Lisa Barnes on this to try to come up with a small version of social determinants of health questions. These are really in their infancy, widely open for discussion and debate, trying to focus on economic status and stability, access to healthcare, discrimination, neighborhood home safety, transportation access, and social contacts. 2 questions on diet and exercise. Again, we hope that there will be, outside of that nucleus of the core UDS, that there will be optional forms that may be able to expand on this, potentially based on current U19s that are studying social determinants of health, because what's most important is that there is strength in numbers, and the more centers we can get using the same data collection instruments, the further we can potentially take our data if those data collection instruments are validated and advance the science. Form A2, co-participant. This has largely been focused on relationships of the study partner to the participant, as well as who is the study partner. And again, we really feel that this information could be collected by whatever means is best for the center, lightening subject and or site burden. There was some thought on this, that what we're really trying to capture here is information about how reliable the informant is. Do they really know this person? How much time do they spend with them? What is that? The modality of that, is that all, FaceTime? Is it a telephone call? Do they live with them? And that this was much more important than whether or not the informant was a male or a female, or what their years of education. Again, there's a lot of interest in caregiver research, and we certainly hope that there may be optional forms that go beyond this, but for the core UDS, let's really focus on this. And we've not done a really good job on this in the past, looking at the UDS forms. Exposure type and frequency of contact has not been collected traditionally, and this is critical. We would collect this at any clinical trial. We want to know that the information that we're collecting on that participant is worthwhile. So these were the primary changes. For Form A3, this is our family history form. This is asked in the past a lot of different questions about what gene was your mother or grandmother identified with. Many of our participants may not have that information, and even if reported, it may be less than reliable. Again, this is a form that may be collected through a variety of different measures, because it's asking a question. So the focus really is to capture family history and first degree relatives. Capturing this in more extended family trees may be less than important, and also recognizing the resource that we have in NCRAD, ADGC, and the AD sequencing project. They're really defining genetics that are objective, and that data can be linked with our participants. More important for us to know the familial risk than to be asking for site-specific mutation status. And so Liana Apostolova, leading this group, has really done a wonderful job with kind of streamlining it down to the level of questions that our participants can really answer truthfully, honestly, and hopefully in doing so reduce site and participant burden. With that, I'd like to turn to Cindy, my co-chair in this group, to run through the forms that really focus on, all right, we've got the participant here, we understand who they are. Now, what are their medical conditions, comorbidities, risk factors for disease? Cindy?
Thank you, Greg. I just want to thank Greg as well for all his work he's put in. He developed our NIH-like platform for reviewing the forms and really helped us develop a schedule and keep us on task. So I want to thank him for all of his efforts in leading this group. I also want to acknowledge too, a question was raised with these forms. Have we vetted the different ways they could be administered? So again, could it be done ahead of time before they come in, either on paper, electronically, or self-administered versus clinician or clinical staff administration? We have not yet. And so those are some ideas that we'd love input on what forms could we implement more readily, or what kind of studies could be designed to see how reliable they are across different modalities. So thank you for that comment. So Greg is circling his pointer there. So the next forms we'll go through. So A4 is the form that collects participant medications. So it's pretty straightforward. What NACC has done is the form currently has the top most prescribed medications as of the point when it was created. And then if you could click next, Greg. Let's Greg, if you could, oh, maybe it's just, there you go. So there's also a search tool though. So it has top most prescribed medication, top 100, has a list for people to put down commonly prescribed or commonly used over-the-counter medications, and also a lookup tool. And so if you can go to the next slide, I think as a group, it was pretty straightforward. We looked at the evidence from NACC, how often this is used, how many people are on medications, prescription medications, and obviously we know that medications could have a huge impact on protection, but also on risk factors or other modifying effects of risk for Alzheimer's and other forms of dementia. So we knew that medical data was critical. We also recognized that the current NACC form does not allow for dose and duration to be fully captured. And so we looked at different types of medication capturing software. And given that NACC is moving towards using REDCap as its database, that many centers are moving towards using REDCap for their data collection, we thought that it'd probably be best to have something that could be, instead of just changing the form, which could be done, we could update the top 100 medications, keep using the lookup tool, but also try to integrate it with some type of software that would allow for electronic data capture, integrating things such as access to Epic or other forms of electronic health records. So the goal would be to move towards something that would improve accuracy by allowing linkage to other forms of software and electronic medical record capture. So that was a general recommendation. Again, specifics have not been identified, but this is a general thought. Next slide. So the A5 and D2 form, as many of us know, were separate forms that looked at medical health history. And so those forms, one was supposed to be subject reported and one was clinician assessed. But for example, somebody having a thyroid disorder, usually that's obtained by history, looking at their levothyroxine dose, etc. And so what we decided to do is to collapse these forms into one in order to streamline things, reduce redundancy because of that significant overlap in questions. And then again, there was this usually again, the health history is collected by a combination of factors. If you have access to the direct medical record, of course, that was preferred to be used, but some of it's collected by subject history or participant history. Some is collected by co-participant input. And so what we do is we're moving toward trying to use the highest level of accuracy for the medical data, but recognizing that it's going to be a compilation of ways that we collect that medical and other historical data. One note, as I just slipped there and said, we're moving towards all the forms saying the word participant instead of subject because the importance of viewing this as an engagement with people as opposed to something we do to them. So all the forms will be changed to use the word participant instead of subject. The other thing we recognize as we think about using these data on a larger scale is that there were a few items missing from comorbid, let's say, so for example, some composite measures. So one example that came up was, do we have a composite measure of other comorbidities? So things such as the Carlsson index. And again, we're not wedded to the Carlsson index, but just as an example of being able to capture the heterogeneity of disease that people may have in the context of their other neurodegenerative processes. And also cardiovascular indices such as the American Heart Association atherosclerotic cardiovascular disease 10-year risk percentage. Next slide. So in general, what we did was revise the forms. And again, we'll go into this in more detail in the sessions, but what we tried to do is to capture these important medical, psychiatric, and neurologic comorbidities. We recognize these are extremely important. And we want people to use the most reliable information to gather that information. So again, if you can use medical records, great, and corroborated by the physician or clinician in the team, but recognizing that we may have to depend on reports by participants or study partners at other points. Next slide. So again, continuing these recommendations. So again, we'd like to combine these forms into one. We'd like to use some branching logic. So again, yes or no questions that would then open up other additional questions to try to make it more efficient to collect the data. And again, this might be something we could have people complete ahead of time so that they can ask questions or remind each other about what their medical history is. And then we've refined the questions to really better capture the severity, the timing, the impact of these various conditions on cognition. We did add or proposed to add a question on COVID infection and severity. So again, hearing from our presenters earlier today about the importance of COVID, and these are open to being refined as the field's rapidly evolving. And then we also regrouped, there's some questions on substance use. We regrouped those and refined them to try to get them up to align better with some other research being done in substance use disorders, and also to add in, with the increasing use of cannabis, to add in some questions on cannabis use. Next slide. Also, because head injuries can be in the context of not just traumatic brain injury, but also less head injuries that maybe not termed traumatic brain injury, we've gotten some input on how we can better refine some of these questions to have them more aligned with other research that is ongoing, looking at the impact of head injuries on cognition. And then allowing for a few questions to allow completion of a comorbidity index. Again, we're not wedded to Carlsson. There's different forms of it, but something that would allow some type of compilation of comorbidities, and also for vascular health, making sure we have all those elements as well. And it's been mentioned a few times, we would really like input from geriatric psychiatrists on the questions on psychiatric comorbidities, and again, how we can better capture the severity and streamline it. So really focusing on conditions that either precede dementia, contribute to dementia, or are a consequence of dementia. And then we haven't gotten to this yet fully, but again, somehow to have a form that we can review, or either on the computer or in hand, review with participants at each visit, asking them, does this look right? And then adding only things that are pertinent so that we don't have to keep going back and asking them each time to make sure that things stay streamlined. Next slide. Again, I think I mentioned before, but Amy's posted in the chat where you can access these forms. You can review as we go through these. And again, having those handy for the breakout sessions will be really helpful. But the breakout sessions will have these forms to show you as well. B1 was called the physical form, and as an internist and geriatrician, I didn't really see it as a physical form. It's more of a vital sign form. So again, really B1 just captured whether they had vital signs done, and then also whether they had hearing or vision problems. It didn't test hearing or vision, but just asked if they had problems. And there were no other physical exam components included, as the neurological exam was on a separate form. There, as Nina mentioned this morning, there is a new blood pressure module that was recently developed and implemented. So we wanted to be able to capture that and integrate that work that's been done into the new forms as well. Next slide. So again, we propose that this form be relabeled as vital signs and anthropometrics to really reflect what the data is that's being collected. Also to change units to metric to avoid errors in calculating things such as body mass index from height and weight measures. To integrate the new blood pressure module into this. So again, that also includes instructions on how to standardize measurement of blood pressure. And then also consideration of adding waist and hip measurements so we can get a better sense of central adiposity and how that increases metabolic risk for dementia. We talked at length about what to do with the questions on hearing and vision. And really at this level, they're more so quality checks for whether someone's hearing and seeing well enough to do the cognitive assessment. So that's what we decided and recommended. Again, we're open to suggestions, but moving those to the neuropsych testing so that the team members who are administering the neuropsych tests can note were they hearing well and seeing well enough to accurately complete the testing. Next slide. Now, for the neurological examinations, this is Form B8. Again, this was reviewed by 3 of our neurologists on our task force in detail. And again, there are some suggestions for changing, but this is an area we really would like input. So again, this is one that's not as well developed because there's such a need for input on how are we going to use these data. So currently neurological exam signs are grouped under a diagnosis. So for example, this person has physical exam features suggestive of normal pressure hydrocephalus, and then underneath that are listed some of those features, supporting features. And so it was thought that by listing them under a kind of a group diagnosis, it may limit the description of some phenotypic heterogeneity across different pathologies within the same individual. Also, as noted previously, the physical exam did not include cardiovascular exam or other relevant physical exam findings. And again, the clinician level of experience, such as are they a fellow or are they a faculty staff that's been doing this for many years, was not noted. And also, has this clinician seen this participant before, and are they familiar with the prior assessment? So there's some quality checks that we thought should be addressed. Next slide. One thing that we're really thankful for is that NACC supplied us data for all the different forms that we reviewed. They supplied us data on how often they were used, how often they were completed, what kind of the findings were. And so in the neurological examination, we found that this is very helpful. So for example, from the NACC data, they found that the frequency of findings for Parkinsonism was about 10%. And you can see that the questions that we had looked at findings being consistent with stroke or PCA or PSP, ALS, and PH were very, very infrequent. And then there was a group of about 20% had other findings. So again, these were usually free text. And so we weren't really sure how are these data being used, but there's obviously 20% of individuals who had other findings. Are they important or not? We can't tell. Next slide. Next. Thank you. And so, no, I'm sorry. I meant to advance the animation. Thank you. So another note was that some of the findings from B8, which is the neuroexam findings, did not align with the diagnoses provided in D1. So for example, for those who had posterior cortical atrophy, about 27% of them didn't have any physical exam findings supporting that diagnosis. And same thing with FTLD with motor neuron disease. There are about 30% of people who had that diagnosis but did not have any physical exam features of ALS. So again, our neurologist team members thought that, again, how are we using these data and how helpful are they? So for this, the NACC follow-up packet, so again, how are these physical exam findings helping us capture progression? So in this case, Parkinsonian signs in the table at the upper left here, about 85% did not have Parkinsonian signs, 3% or about 4% did have signs, but again, as far as how often they progressed, if you look at still within that left upper table on Parkinsonian signs, you can see that there's about 5% that show progression, but also about 5% that are random. So again, we know that disease does kind of ebb and flow, so there may be some participants who did have symptoms one time, didn't another time. But again, it could also be reporting error and lack of standardization. And similar findings for cardiovascular disease or cerebrovascular disease in the table to the left on the bottom. So I just showed the graph on the right because you can see on the x-axis, these are all the different physical exam findings noted. You can see that consistent, which most of those were negative, in the blue, which means that most of these things were negative, but there's some, the orange shows some progression and the yellow shows that it varies randomly. So again, whether it varies randomly is a true fluctuation in the disease or something else is not clear. So again, that's why there's questions about this form. Next slide. So again, further issues to clarify, how are we going to use these physical exam findings just internally to confirm a research diagnosis, or as individual raw elements that can be used for analyses or AI data, as we've heard about today? And then how do we best capture variability across sites and improve consistency across sites? Next slide. So again, we recommend that we find a way to document the clinician level experience, familiarity with participant and prior assessments. Also, because of possibly moving towards more video-based assessments, assessing whether it was video-based or in person, which may also impact whether it was a focused or in-depth exam. Using a gateway approach, so yes, no, to group physical characteristics so that we can, again, kind of assess these variety of symptoms and signs that may be noted on the exam, how they relate to heterogeneity of disease. And then other pertinent physical exam features, such as a cardiovascular disease, neck circumference, so just part of stop bang and assessing sleep apnea risk. And then again, psychiatric features. We don't really have a full mental status exam other than our cognitive data to really assess if there are other physical exam features that are important. But again, how are we going to use these data? Next slide. So as we looked at the physical exam form, the other thing that was thought is that we should have a little more guidance within the form itself. While there's a wonderful guidebook, most people don't go to the guidebook and go to a separate form. So either having links or somehow having a way to standardize it. And then I think Doug's question was a little prompt here for consideration of adding other things such as adding back UPDRS, timed up and go, smell testing, some more objective measures. And then as we'll hear from the digital group in just a little bit, anticipate addition of digital measures in the future for some of these physical exam findings. Next slide. So the B9 clinician judgment of symptoms, again, this form is really designed to help us understand what is the trajectory of this person's course of cognitive decline, behavioral decline, and motor decline. So there are features in each of these domains. But again, coming back to how do we best capture the onset of these different features, what was a prominent feature, but also time course of each of these different features as we look to explain heterogeneity. So currently the forms do a wonderful job of assessing by the clinician cognitive, behavioral, motor domains, symptoms and sign within each. When it was first recognized in the prominent domain it was affected, the participant and co-participants' input, though, is only captured as a decline in memory, so not other cognitive domains, behavioral and motor domains. And then this also is a question that leads to the modules in Lewy body disease and frontotemporal lobar degeneration. So next slide. So again, I think we all recognize that understanding how these clinical progressions evolve is extremely important. So we wanted to keep those features. Next slide. But again, what we'd like to do is to make sure that we're getting input from the participant and the co-participant and the clinician on all these aspects, the cognitive, behavioral, and motor domains. Not necessarily all the specifics from the participant and co-participant that would stay with the clinician, but at least getting their input on whether or not what the key features were. And then also, again, we'll be moving towards not having these additional modules. We'll be moving towards letting the consortiums and U19 grants look at these other disease entities, but try to move some of those key features into the UDS core component in clinical assessments. Next slide. So I think I'll turn it back over to Greg and he'll go over D1. Again, remember to bring your questions to our breakout groups after the end of this session.
Fantastic, Cindy. Thank you for going through those very complex forms. We'll try to wrap up. Form D1 is really the last form that we'll discuss today. This is really a crux, and I just want to have a call out to Jeff Burns from Kansas and the working group on this form. This really is perhaps the most used and useful data element in the entire UDS, besides maybe age itself anyway. But this form has been useful for us, but on our review, it was found to be somewhat scattered. The form proposal is to reorganize this into two discrete sections. One section that will capture clinical diagnostic criteria. Does this person meet criteria for possible probable AD, or do they meet clinical criteria for DLB? And then The second portion, Form D1B, will incorporate then the biomarkers that we're all collecting and that we know are critically important. We plan to use branching logic on this, again, so that every question does not have to be filled out. If you don't have an MRI, you will not have to answer 7 questions as to whether or not the MRI showed evidence of this, that, or the other thing. You'll be able to just simply say, I don't have an MRI, or a PET scan, or spinal fluid, or blood-based biomarkers. So if there are no biomarkers and you're observationally following a patient clinically only, there's a few simple checks. It's a very easy form, reducing site and participant burden. If you have biomarkers, you only need to provide information on what you have. The advantages of the new form as well has been really broadening. A lot of the work that the subgroup has done is broadening the clinical diagnostic spectrum, cleanly separating clinical diagnosis from biomarker-driven diagnosis, and reorganizing these biomarkers again by modality. So the summary of recommendations, again, in terms of the clinical diagnosis portion of it, is actually to ask. We encourage, and the NACC instructions have long been, use all the data and the best data you have. But we do recognize that because of the logistics in every individual center, not everybody's doing it. We do want to be aware, was the neuropsych cognitive battery data used to inform the diagnosis, or was this a clinician only diagnosis as well? And hopefully we will move towards more uniform approach to filling out this form, given that diagnosis is one of the most critical variables. A lot of interest, discussion, and push in the group to add behavioral presentations to the MCI section. We think that we've been disregarding this. This isn't just for frontotemporal dementia. We all know that sometimes our Alzheimer's or vascular or Lewy body patients actually present behaviorally before they present cognitively, and we've not been capturing this well. Extend primary dementia syndromes to include other conditions that aren't on the existing form. And then as we move into the biomarkers, of course, this form 1B as proposed has 2 sections, biomarkers, imaging, and genetics. Again, really asking by modality. Do we have MRI? Do we have FDG, PET, DAT scan, genetic tests, or molecular biomarkers, CSF or molecular PET? Again, and using branching logic. If yes, you have that, then there'll be a question that will ask you, did it indicate a diagnosis or not? If there's a negative response, you're done. No, we don't have an MRI. You don't have to answer anything else. And then section 2, at the end of the day, combining the clinical diagnosis with the biomarker diagnosis. What is the best guess at the etiologic diagnosis as well? And this is important. It allows ATN, and as biomarkers are accumulated, and we can already use things like DAT scans for Lewy body or Parkinson's dementia, future ATN positive growth. There are inclusions on the biomarker forms for thinking about blood-based biomarkers. We understand that these are works in progress, but again, with an eye to the future, what's this going to be like in a few years? Capturing that data now we think is important, not focusing like with Form A3 on what is the specific genetic mutation or what is the absolute CSF A beta 42 to 40 ratio, knowing that the assay methods and the normative values may change from site to site. In these forms, only based on the site's experience and normative values, is it positive or negative? We really do hope that the ADGC and NECRAD, the Imaging Steering Committee, biofluid steering committees in the future will create data streams where we will have the actual data in a way like Centiloids, where we might be able to compare apples to oranges. So we know that you guys all have many questions about this. Again, I hope we've stressed, Allan, Cindy, and myself so far, that this is really a starting point for constructive discussion. We've done a lot of work on this, pre-vetting a lot of issues and trying to find some common ground, but clearly, as you can see from the presentation, and for those of you that have pulled up the forms and reviewed them, know that this is, again, something that we're hoping to refine. Please keep in mind with your suggestions, the science, as well as participant site burden. And please try not to think only about your site, but to think about how we can leverage the entire ADRC infrastructure to address these scientific questions, because that's been at the forefront of everything that we've been thinking about. Remember, this doesn't constrain you. We want you to be able to do your own things outside the core UDS. We want to keep that core UDS tight so that we have that ability. We've not yet worked on, which forms are optional versus mandatory. That's something that can be brought up for discussion at the breakout sessions as well. So for the clinical measures diagnosis group, we've really set up those breakout rooms in 3 different areas, forms A1, 2, and 3, in terms of forms that are really talking about who's the participant, what's their background, what's their family history, can we trust the informant to be providing reliable history, or the co-participant rather. Forms A4, 5, B1, and B8 tell us about this person's health history, their medications, their risks, their physical neurologic exam, and B9 and D1 we've clustered because we think that these are really synthesis forms that are bringing together that information. Of course, remember, there will be breakout rooms for the cognitive working group led by Andy and Lisa that will be speaking shortly, and the tech work group led by Rhoda and Jeff K as well. In total, 5 different topics. There'll be 2 discussion groups for each topic to make sure that the numbers of attendees are small enough that all of your voices can be heard. Remember, it's a team effort. If we're climbing Everest, let's do it as a team. Let's make it to the top. So I know my slide says next up is the cognitive working group, but I think
it's me.
It is
Rhoda.
It is Rhoda.
I think it's me.
All right, very good. I thought I had a mistake on my slides there. I apologize for that, Rhoda. I appreciate all of you out there, and I hope that you stick around and really participate in those breakout sessions.
All right, let me see. I wanted to. Okay, so I'm going to try to power through because I know that we have a hard stop at 4 and I want to make sure that we get to the cognitive group, because the one thing about the technology work group is we actually don't have previous forms to deal with. So in fact, I'll see if I can just sort of power through this to give the cognitive work group enough time to move forward on theirs. So, the charge here is, to shift from UDS 3.0, which for us, from the tech standpoint means we have to look at what's already been done, but then we really need to think about how we're going to bring technology into that. So, it turns out though, when we first started out as a group, we just decided, see what's already happening. And it turns out that there's actually already a lot of work being done by the ADCs across the country. And so this is sort of an example of just a summary of some of the surveys that comes through where we see that a lot of ADCs are already using digital technologies, and there is vast interest to continue doing so. So, just to give you a very quick sample, there's all sorts of technologies that's being done. It's very hard to quantify very simply everything, but this gives you sort of just a snapshot. And so really, what we were tasked to do is to really figure out how do we move forward then with digital assessments, both within the clinic and then out of the clinic. And so this is the task working group, and I get the privilege of sort of representing the hard work that this whole team has been doing. So we really wanted to separate this idea that there's what you can do within the clinic. And the reason we wanted to separate the two is because within the clinic, you don't have to worry about things like a participant's connectivity. So we thought that this is sort of a lower hanging fruit for all ADCs to pursue. And what we wanted to do is just give you a sampling. So this is not by any means comprehensive, but just to give you an overview of some of the work that's being done, particularly through the task force members themselves, because these are ones in which there's already, protocols, consent forms, et cetera, but we're very open to other ideas as well. So one of the things to realize is that there are things you can do with what you already have. So for instance, you can take regular paper pen tests, and because things like smartphones with cameras are so ubiquitous, there's actually the possibility where you can take a paper pencil test and then convert it into a digital image. And then using machine learning capabilities, you can upload the image and analyze it. And so I just wanted to give you a quick example. This is some work that some of my colleagues have done at Boston University. These are my machine learning colleagues, Vijaya Kolachalama and Yanis Pashalidis. And they were actually able to take the digital image and then look to see if they could determine, just based on this digital image, whether they could differentiate those with and without cognitive impairment. And what's interesting is they tried different models, and they found that actually just with age plus the digital image analysis, they could actually get to the same level of predictivity if that was similar to if they added additional variables. So this is just a very quick overview about where you could even take what you already have and start to digitize it. Then another example is the Linus Health platform. We're deploying this within BU right now. And I just want to see if I can do this, sorry. I want to just give you a sense. It's a platform, so it provides cognitive assessments, questionnaires, does medication tracking, et cetera. And so just to give you an idea of what this looks like, oops, sorry about this. Let's see, oh, there we go. This is just to give you a snapshot of how this application, it can be done on a tablet in your clinic. And you can see some of these tests, they're actually pretty easy to do. And then let's see. These are some, speeded responses. And then some naming where you can do some voice recording, which as many of you know, I'm very enthusiastic about because it's a very big cognitive task. You can do questionnaires, et cetera. And then just to give you another idea, here you can, I'm not sure if you're going to be able to hear this, but I'm going to hopefully try. Let's see. Looks like it's not working quite the way. Oh, there we go. Sorry.
Before we start the test, let's practice with the pen. Copy this line as many times as you'd like. The pen can be used to write and draw like a standard pen. You can rest your hand on the tablet when using the pen, just like when writing with a standard pen.
So I'm hoping you're hearing
When you're
this.
done practicing, tap the next button. On this test is a series of lines. Start at the blue dot. Then trace the line in the direction of the arrow. When you are done, tap the Next button.
So this is just to give you an example of how simple some of these applications are actually to administer, and how these applications are really set up to make it easy for even less trained staff to be able to do. So I'm just going to move on because we have, in addition, the Linus platform is actually agnostic to tests. So it can actually take, sort of your favorite test and bring it into the platform. So TabCat is sort of one of these applications that was developed by Kate Posen at UCSF. And they've done quite a amount of work validating sort of their digital platform. And this can actually be integrated. So, they've been able to demonstrate sort of how effective it is in terms of establishing A-beta, differentiating between those with A-beta PET status, and they've actually done a number of other tests as well. So the point here is that, we can take any of the different cognitive assessments or behavioral assessments that you have and add it into a single platform so that it looks like a much more unified assessment. In addition, we're very interested in sort of looking beyond, sort of more standard cognitive tests and really looking toward things that are a little bit more agnostic and that are more culturally language fair. So for instance, at BU, my colleague Lee Goldstein has actually developed an optical capability where he can measure amyloid in the lens. And this is obviously an alternative to doing much more expensive PET imaging and still getting the same type of biomarker. In addition, Allan Levey's group at Emory, they've developed the VisMet, which is a visual memory eye tracking task, and where they are able to track by moving objects in and out of the screen, they're able to track the eye movement. And from that, they've been able to show that this is a really effective passive actually assessment that is culturally, language, age, education, agnostic, and able to differentiate sort of between those that are cognitively intact with mild cognitive impairment and with AD. In addition, there was gait and balance tools that you can bring in. So these are just some of the options that are out there that have been used. I noticed earlier someone was asking about these timed up and go tasks, which are very easy to do with these digital metrics. And similarly, there are other options for how to do this. So that just was like a really quick speed through in terms of what's possible within the clinic. And what I wanted to do is actually focus also on what can happen outside of the clinic. Because that's where I think digital technologies have opportunity to allow us to continue to do things. And we've had a lot of experience now of that with the COVID situation. So again, we're just going to highlight a few things here. So one, earlier, I think yesterday, they actually mentioned the e-consent work that's been done with the Emory team as well as the Wisconsin team. And this really allows you to, get informed consent using some remote applications. This is work that has been done with Sage Bionetworks, and there's lots of opportunities to take this as a framework and start to push this out into a much more robust system that could be used across all ADCs. In addition, the Wake Forest team has been using the GrandPad because they've been worried about how do you do these remote assessments when maybe some of the participants don't have access to the actual technology. And so there's a lot of value in being able to then extend your ability to do questionnaires, to do testing in general without worrying about your participants' connectivity. But there is, there are some limitations, particularly GrandPad itself has some limited features, although they are willing to work with you if you sort of have a larger ask that they can serve. Again, the Linus Health platform that has both in-clinic capabilities, also has remote smartphone-based capabilities as well. As these are same kinds of tests that you would give in the clinic, you can give on the phone. They have, paragraph recall. In this case, you don't actually have to be able to read it. It can actually read it to you, and then you just retell it. Again, this is an opportunity to push this out into to the remote environment, do assessments on a more frequent basis. We can add in questionnaires, but always, and this is the case not just with this platform, but any remote platform, is you do have to worry about accessibility of participants to these technologies. So I wanted to sort of pivot a little bit and really talk about, what do we think about when we're talking about innovation? So in research, we kind of go where everybody else goes, and we think like, oh, we need to think a little bit out of the box if we're moving toward innovation. And I always like to sort of say, well, what happens if it's not a box? What if, we really need to open our minds to thinking about things in a very different way, and particularly when we come to something as complex as the brain. So I always like to make the point that technology is not supposed to be simply doing a better version of what we've already been doing. Technology is actually supposed to help us make a very giant leap forward. And I think that that's really where we need to think about and push beyond our comfort zones as we start to think about how do we really capitalize on what technology can do. And so sort of the person who's really leading this, and has been actually for over a decade, has been Jeff Kay. So I'm actually a very poor substitute to describe Jeff's work with the CART platform, where he's been able to embed, within the home, within mobile, and across different kinds of digital data streams to collect data passively ongoing in people's homes. So this is just sort of a visual of how some of these sensors that he embeds in people's homes. And he's been able to collect this data longitudinally over time and really gain great insights that our current methods of doing in-clinic assessments don't allow us to do. And he will be leading one of these breakout rooms where he can talk much more effectively in terms of what he's been doing on that front. Now, so there's a lot of different value. I don't actually want to go through this because I know we're running short a long time, but he does have a technology agnostic that also allows sort of integration and customization. What we would like to highlight further is that he also has the capability in which to use mobile devices, kind of go wherever you go. He can sort of embed a lot of these questionnaires that we've been talking about on and then collect additional metadata in the background. And so this is just some examples of some work that he's been doing. Again, we're a little short on time, so I'm not going to go into huge details about this. But I did think that this was very interesting, that these are sort of the kinds of information that he can pick up through his various applications through CART. And I think it's very interesting to note that even within low-income participants, how he was able to track changes in behavior due to COVID, and that's what these graphs show. And he had already embedded these sensors to allow him to do this. So I'm going to sort of try to really quickly now summarize, though, this kind of pushing the boundaries forward even beyond what we're used to. And I really like to talk about the concept of digital biomarkers and really thinking about this in a very new way, and realizing that we're going to have multiple digital data streams. We're going to push it through our data science AI capabilities. And we're actually going to develop, I think, this idea of different signal patterns. It won't be the same. So we have to rethink the concept of biomarkers. It's not going to be the same signal. It's not going to be consistent. It's going to be very dynamic. And it's going to be dynamic within every single person on an ongoing basis. And I really think that that's what we need to think about as we're considering the concept of digital biomarkers. And once we sort of redefine it in that way, then we can work with the FDA to come up with new regulatory standards rather than relying on current gold standards, really moving this forward in terms of establishing new gold standards. And once we're able to do that, then we may enable the opportunity to potentially do things like virtual clinical trials. And then obviously, ultimately, we're trying to get to care. And so I'm just going to end here with just this thought that as we move forward, I really think we need to not be prescriptive in who we think that we can work with. I think we need to take all comers, and I do think that together we'll be able to solve our problem. So I'm going to end with a big thank you. You've heard repeatedly how many people this has taken, and our group has literally intersected with all others. And just a reminder that following the cognitive group, we will have these breakouts. So I'm going to end there.
Can you hear me?
Yes.
And can you see my screen?
It's in edit mode, so if you can
Okay.
go to presentation.
No matter how many times we do this, right? And turn my camera on somehow.
That's it.
Okay.
You got it.
Okay, perfect. Thank you. Okay, so thank you, everyone. I know we are really behind, so I want to move through this quickly. Are you seeing the actual slides though, or the presenter mode? What are you seeing?
We see the actual slides.
Okay, okay, perfect. Okay, so I want to echo everyone else's acknowledgement of the Clinical Steering Committee and of course, Allan for his great leadership. Andy and I have the task of just introducing to you what our charge was, and that was the cognitive battery. So these are the cognitive work group members. And in addition to the pictures, these were the people who were appointed in 2019. We also had some great partners, Sadeshna Das, Hiroko Dodge, Yorgos Tripatis, representatives from the CTF, NACC, and NIA, and a number of invited guests that you'll hear about shortly with Andy. So, you know, we had some general considerations when we thought about our battery. And, you know, we knew we needed to keep the total cognitive testing down to 1 hour or less, preferably less than an hour. The current battery takes about 40 minutes if you stick to all the UDS tests. We knew we wanted to optimize the paper and pencil test to allow time for novel digital computerized measures, which you just heard about with Rhoda. And we wanted to plan for at least 2 tests in each domain, pairing one conventional test and one digital measure. So you've already seen this before at our last meeting, and also Allan nicely went through it, so I'm not going to belabor the point, but I did want to just point out 3 things that were important for us for cognition in particular. And that was, you know, we know that we have, you know, rich cognitive data. So we want to be able to maintain and enhance the value of the longitudinal data that's already been collected. We want to really minimize participant and staff burden. We've heard you that we ask people to do a lot of things. So we keep that in mind. And we really wanted to be able to select tests that would be sensitive for early detection, longitudinal change, and comparable across diverse populations. So I'm going to turn it over to Andy so that he can go into more granular detail about what our actual decision-making process was for our interim recommendations. So Andy.
Thanks, Lisa. And I think we can catch up a little bit. So first off, as you've heard, the intent was very much to make this a data-driven presentation. So, you know, I can't thank Gary Chan enough from NACC, who very quickly did some major analyses in order to give us a first look at this. And we'll just touch on a couple of these major findings, but the final data will be presented at AAIC by Gary this year. So anyway, thank you very much, Gary. So there's 2 points here. One is looking at the criteria of sensitivity to longitudinal progression and using the CDR as independent from the cognitive data itself. And then the comparability for race and ethnicity and language. Okay, next slide. So there's 3 of these data slides that are that Gary prepared that are set up basically the same way. The first one looks at 3-year progression by CDR, looking at the area under the curve for the ability of each of the cognitive tests in the battery. Maybe a little hard to see, but you can see the overall pattern. But looking at the ability of each test to predict progression 3 years later by CDR. So the panel on the left is going from cognitive normal to MCI or AD, basically de novo development of impairment. The panel on the right is from transition from MCI to dementia. And maybe best to look at the overall area under the curve for the global composite for the UDS3 battery. You can see at the bottom, they're running about 0.8 for both of those. and would be obviously higher going all the way from normal to dementia. So number one, for progression, the test battery overall and most of the individual domains are sensitive to progression, which is one important feature. Next slide. The next two panels look at the effect of race, ethnicity, language. So this is a snapshot of baseline effects. And essentially what Gary found was that there's a significant black-white difference and testing in Spanish versus the white population effect, where those scores are uniformly lower. So that's at baseline. Now go to the next slide. Now the key really is what about progression? It turns out if you account for that baseline difference and then go to looking at sensitivity to progression over 3 years, the effect, mean effect is almost zero. That you see the falling right down the dashed line by and large, especially for the black-white comparisons, which is by far the largest. They're not, the sample size, which I don't have exactly here, is not that large for the tested in Spanish versus white participants. Anyway, this gives a first snapshot. Much more detail will be forthcoming from Gary at AAIC, and I'm sure a nice report on this. But go next slide, please, Lisa. But essentially, we can conclude that looking at the actual UDS3 data, as enthusiastic as we all are to go into the future and get to the UDS4 battery and so forth, it's really critical to look at the large investment in the UDS3 and understand what the performance characteristics. So this is just the beginning, but it does look like the UDS3 is performing reasonably well. Now the question is, how can we improve on it? And a lot of the criteria for potentially improving on it, I think, has been discussed. So, we'd like to leverage technology, as Rhoda just spoke about. We want to have good psychometric properties. I don't think I need to list them all, but you can see them on the slide here. We need the test to be quite applicable in diverse populations, potentially translated or translatable. And there are relative degrees by which different instruments are even translatable if they haven't been yet. So those are all considerations. Validation studies, you heard about some validity studies with regard to biomarkers and outcomes, but much more needs to be done in that regard with clinical outcomes, biomarkers, and ultimately neuropathology. And then there's a lot of considerations in terms of making sure we have anchor items to be able to harmonize to other batteries from even at up to a global level, so that those hundreds of thousands of whole genomes that'll be sequenced that Dr. Maslia spoke about earlier today will ultimately be something where we have quantitative phenotypes to do those association studies. I think we could even discuss during the breakout, should we have alternate forms versus just longitudinally model practice effects with repeated testing on the same instruments, the potential for adaptive computer-based design strategies and branching logic and so forth to make use of, to move ourselves into the future, as Rhoda was talking about. All these are possible and important to look at carefully. Next. I think you actually heard a lot about relative to the progress to date. We've been looking at a lot of the instruments since the current UDS3 cognitive tests are going pretty well, and we now have the TCOG telephone version and preferably the VCOG to do it by video. We now are looking at some of the technologic solutions that are out there, the tablet and mobile-based measures that are on the slide here. And there are other ones that we're in various stages of learning about and considering, also looking at what pharma is doing, because they're extremely concerned about power and sensitivity to be able to detect dynamic changes that are associated with treatment. That's probably the #1 priority there. And we really welcome your suggestions, starting in the breakouts, but also please email us or otherwise communicate things that you're aware of that you think really meet our criteria that we should be looking at. Next. So the interim recommendations are essentially to continue with the UDS-3 battery for the moment, and especially with the relatively new TCOG and VCOG remote assessments, because those continue to be important. Even beyond COVID, they're likely to be important for many applications. And to collect data so that we really have a good sense of the in-office versus remote performance, to identify the best performing instruments and be able to move on from there. Another topic is one of standardizing verbal learning. Given how important memory is, at least for canonical Alzheimer's disease presentation, we're all using different verbal learning tests. There's 4 or 5 different ones across most of the centers, at least. We have a recommendation to standardize, but another approach is potentially to just harmonize them post hoc, but it's not the same as having the same instrument across centers. It could potentially, if there is a consensus, we could do that. We could potentially have one of the newer digital measures, and we'd like to hear your opinions on this. Rhoda's already talked about digital speech capture, which we have consensus on and support, and potentially digital pen or other digital capture. We should make the most use of that technology. So we need to complete the selection process and analysis and continue with looking at the existing UDS and then especially the TCOG and VCOG data as it comes in, and then to design pilot studies for the newer selected instruments that maybe groups of centers would want to execute together. Next. So for the breakout, we'd be interested in your thoughts about verbal learning and paragraph recall. What kind of a verbal memory or verbal learning consensus test would be optimal? There's, we won't spend a lot of time on this, but and Rhoda's already talked about digital assessment, but for the next one is really the MoCA issue, which has been published in the New York Times and Washington Post and elsewhere in recent years. But should we be thinking about an ADRC NACC screening instrument that we would have more control over? Should we continue with the MoCA, which seems to perform pretty well, as in the slides that I just showed from Gary Chan? It's up for discussion. What about sensory function? Where should that go? Is that just part of the screen? Should it be part of the battery? Where does it fit in Greg Jicha's atomic orbit? Maybe we'll talk about that in some of the sessions. And I think with that, it's time to move to the breakout sessions. Are there any instructions that we want to provide?
So I appreciate that, and we'll try to move forward. I appreciate Rhoda and Lisa and Andy and the way all the groups have worked together. We'll kind of try to march through this quickly. There is a link to join breakout rooms. This will allow you to select which breakout room you would like to be in, and that's great. And you can return to the main room, in my understanding, at any time that you want. The forms have been sent to everybody and are laid out quite clearly. There are comment buttons, open text fields for you to provide feedback. This is really important because in the future, after this is not the end of this work. This is the very beginning. Each center is going to be asked to submit their comments, and we want each center to come together and present a unified recommendation to the CTF. And you'll use these forms, the current UDS-3 form next to the UDS-4 form, and you'll be able to fill out those sections. You'll understand the reasons for the proposed changes on the left of the screen and provide your feedback on the right. We will host some webinars. I'm not going to go through this very busy form or busy slide for the sake of time, but we hope where there are issues, where there is significant debate, to fully vet those through a series of webinars and engaged discussions before we move on with any recommendations. So join us in the breakout rooms. There are 5 topics that will be discussed, 2 breakout rooms per discussion. 1 breakout room focused on the concept of who's the participant, what are their demographics, what are the co-participant characteristics, what's the family history. Another breakout room topic on medical risks and comorbidities, A5, B1, B8, diagnostic impressions and synthesis in another with B9C1, objective cognitive performance by neuropsychological battery scores, and the technology work group, which is certainly an area that we're all moving into. So there are some global questions. Just keep these in mind as we move into the breakout rooms. We really want to know what's missing. Do the forms capture the appropriate balance between practical considerations and advancing the science, our primary goal? Have we deleted anything that you find essential for research, especially at your center? We may not know that. What do you like? What do you not like? And are we headed in the right direction? Are these forms going to be useful for the overall mission of the ADRCs? What do you envision? Let's look to the future. Where do you think we're going to be in 5 years? Are we going to have to redo this again? Can we have a forward-thinking moment where maybe we don't, or maybe we build a living UDS that can change dynamically over time? Very, very important. And if there are other venues for discussion and or input, we want to hear them. We're vested in your feedback, in you helping us guide this process. The CTF is part of the ADRC network, but the ADRC network and our service to you, our participants, and to everybody out there that needs our scientific advances is paramount. With that, I hope everybody knows if you go back to your agenda, you can click on a breakout session that is of optimal interest to you, and let's talk. Thank you all today.